Just take your time, Philip. It's not so strict.
Yeah, thank you for having me. You guys have an early start.
Yeah. Yeah. Is 8 an early start what you meant?
Yeah, I think so. You must have very dedicated students.
We have superhumans here. Good. Good. All right. All right. All right. Okay, good. Good, Philip. You hear me?
Mm-hmm.
Rồi mọi người ơi, mọi người tranh thủ ngồi dồn lên hàng ghế đầu giùm chị nha. Right. Hello everyone, welcome back to the workshop today. I hope that you guys had a good night last night. Okay, so beginning of the workshop today, we have like Dr. Philip, like we present you the algorithm in nature. Okay, and we can begin before beginning with Philip. Just remember the 3 rules. The first one, turn off your phone or put your phone in silence. Okay. Secondly, don't take video or photo during the workshop. And thirdly, we encourage you to ask questions. Before this is online, so if you want to ask question or answer the question, just feel free to raise your hand to me, then I will like give you like the microphone. Okay, thank you. So now we can begin like get back to like answer.
Hey friends, hey students and everyone. So today it's my great pleasure introducing Philip. Philip, you see me?
I do, yes.
See me yet?
I do. Now I can't though. I lost you, but it's okay.
Okay, now you see me?
There we go. Yes, excellent.
Yeah, now you see both sides. Good, wonderful. Thanks. So it's a great pleasure having Philip. And Philip Campo is a professor at the Computational Biology Department at Carnegie Mellon University, CMU. He directs the undergrad program and serves as the assistant director of the Master in Computational Biology program. And he co-founded the pre-college program in computational biology at CMU. So Philip has been teaching a lot, and probably if some of you has done Rosalind, which you will do in the afternoon. So Philip was the one with Nikolai founded. So this is the guy that makes troubles to you. So of course, you know that Rosalind has been adopted by hundreds of thousands of students around the world. And not only by students, I think, a lot of professors at different countries has used Rosalind for teaching. And I think for the probably not updated information is that the Rosalind has been used by more than 45 countries, and from top places like MIT, Stanford, UCSC, UC Berkeley, and many places that adopted the platform that Philip and Nikolai founded for bioinformatics education. What else can I say? He's also wrote the book in computational biology, an active learning approach that is considered as the best bioinformatics book of all time. And
Certainly top 15.
that's what I can see from posts from social media writing about the book, right? And with that, I think it's my great pleasure having Philip today. And today, Philip will talk about algorithms in nature, right, Philip?
Okay. Sean, thank you so much, and thank you for the warm introduction. Hi, Pavel. I can see you. You only take up about 3 pixels on my screen, but I don't know if you heard me. Hello. So good to see everyone. As Sean said, I teach at Carnegie Mellon here in Pittsburgh, and you may not have heard of Pittsburgh. One of its claims to fame is that in the winter time, when it gets cold, we had a sunset just like the one behind me in my Zoom background today. And we have these beautiful sunsets. And I was here for a few years before I realized that the reason why we have these beautiful sunsets is because we have the worst air quality, the worst air pollution of any city in the United States. So if you have very bad air because of many factories, apparently that gives you very nice sunsets in the wintertime. So there is some benefit to it. I know that a lot of things that you're going to be learning about are going to be based off of genomic study, probably. So a lot of bioinformatics is the study of DNA and RNA and proteins and these sorts of things. And if you check out the Rosalind or the book that I wrote with Pavel, and probably Pavel's talk later today, you're going to get a lot of that. I wanted to talk to you about something a little bit different, a little bit more sort of outside the box that I really enjoy getting to talk to students about, which is the notion of not being presented with data and then designing an algorithm for it, but instead doing sort of an opposite approach, which is looking at a natural system, looking at a living thing, and trying to figure out what algorithm it is using to solve some type of problem in its environment. And this might be something that you have seen before in the context of things like swarm algorithms. So often a lot of students have heard of things like algorithms that bees use for building a hive or solving some problem within their local region where you have a bunch of independent bees that are seemingly behaving independently. But when you zoom out and you look at the swarm itself appears to have some sort of intelligent behavior because you have all the bees performing some task that appear to be happening together, and you have collaboration. I didn't want to talk about that. I sometimes talk about that with students, but I wanted to try and find 3 little short stories for you about how different living things have solved problems in their environment. And I also wanted to encourage you. I don't know sort of what the best setup or what you're used to here, but if you ever have any questions at any point in time, I'm going to try and go slow and steady as I can. If you have any questions, please feel free to stop me. Let me know what you think. And otherwise, I think we're ready to begin. Sound good? Give me a thumbs up if that sounds good. Okay, good. All right. The other thing that's very strange, I should say, about this classroom setup is I've done a lot of Zoom teaching before, but I've never had this circumstance where when you see me, you're looking to your left. But when I see you, I'm like 10 meters in the air looking down on all of you. So I've never had that happen before. So it should be fun. Okay. So the background image here we're going to see very shortly is something called a slime mold that usually feeds on decaying or decomposing matter in its environment. And we're going to talk about that shortly. The first organism I want to show you is E. coli. E. coli is one of the most studied organisms on the planet. And it's the model organism when we study bacteria. So if you're ever going to do a study of bacteria, it winds up being the organism that's studied the most. And one of the problems that actually might be the most studied aspect of any bacterium is how E. coli explores its environment. And I always try to give an introductory analogy of this problem from a book called What If? So there's an author, there's a famous cartoon series in the US called XKCD that are very scientific cartoons. And Randall Munroe, the creator of this, created a book that was based off of a website where he asked his followers to say, ask me a bunch of scientific, absurd, crazy questions. And then what I'm going to do is I'm going to very seriously answer the questions that you ask me. I'll pick the best ones, and then we'll have a serious discussion of these crazy questions. So one of these absurd hypothetical questions that I realized is a really good introduction to this was someone asked him, what would happen if you had 2 people and the people are immortal, so they live forever, and you put them on opposite sides of a planet or on opposite sides of like a continent. So you put them in it's an Earth-like planet. So imagine they're both somewhere in Africa. And so we know they can find each other if they just walk around the continent. And the question that they asked was, well, how long would it take them to find each other? Would it be 100,000 years, a million years? And what approach could they use? And Munroe called his response to this, which I think is a good answer, be an ant, because he borrowed the way in which ants explore their environment. So he says, if you have no information, walk completely randomly. So just wander in different directions. and leave a trail of stones that will point where you are traveling. And both people can do this without any communication among them. And he says you want to walk for a day, and then you want to rest for 3 days. So you have a certain pace that you're traveling. And you want to indicate what day it is on the rocks that you use. And that way your companion will be able to know, when you reach this point. And he says, if you come across a trail that is not yours, well, start following it and start walking faster than you normally would. So you're in a different mode that's like a pursuit mode. You're trying to find your partner. And if you ever lose the trail, we'll go back to your original exploration. The idea here is, if you ever you don't have to come across their exact current location. You just have to find a location that they've been somewhat recently. So because you're moving faster when you're pursuing them, there's a very good chance that you'll find each other. And you might find each other in a matter of years or decades. So he knows a little bit about how nature solves this problem. And I want to show you that I made my cursor really big, so if I need to highlight anything, you can see it. I want to show you that bacteria wind up employing a similar approach that's also based on randomness to find food. So what you see here is, and this is a good reason not to leave cake or anything like that out on your kitchen counter for too long, because bacteria are very good at sensing this is a glucose or sugar crystal. And bacteria are very good at sensing that in their environment and swimming towards it, although some of these bacteria actually miss the crystal. But what they are able to do is turn around and head back towards the crystal. And we're going to talk a little bit about modeling exactly what they do. This is one of the most studied biological systems in the world, because you're just studying bacteria. And there's been a huge amount of work done in knowing sort of how the bacterium works as a machine, as well as what is actually going on inside of that bacterium in order to cause this change on the outside of the bacterium. So the video is showing here is something called chemotaxis, and that's a fancy term that just means that an organism is moving in response to some chemical stimulus. So if you smell a good dinner being prepared, well, you have chemotaxis too, in that it might attract you in that direction. But this is an example of that same sort of phenomenon with bacteria when they have a sugar crystal in their environment. The E. coli itself, the cell, has a variable number of flagella on the outside of the cell. And those flagella can rotate in either one of two directions. So they can rotate clockwise and they can also rotate counterclockwise. It works out that when all of those flagella are rotating, not just in the same direction, but counterclockwise, they all bundle together at the back of the cell. And this, when they're rotating, that forms like a multi-propeller, and that pushes the bacterium forward at this speed of 20 micrometers per second. And that probably doesn't seem like it's going very fast, because that's 20 millionths of a meter every second. That's not very far. But in the bacterium's world, that's very, very fast. So the bacterium, when it's swimming, is able to swim about 10 times the length of its body per second. So if you stack up 10 E. coli together, it will be able to travel 10 of those in 1 second. And that's like a car that travels at about 160 kilometers per hour. So in its world, when the E. coli is hungry and sensing food and traveling in some direction, it's very fast. However, the bacterium will also sometimes stop. So every so often, the bacterium will have one of its or more of its flagella will rotate clockwise. And when that happens, the flagella at the back no longer form this bundle. So you no longer have this propeller moving the bacterium in one particular direction. When you have at least one of them rotating in the opposite direction, it causes the flagella to become uncoordinated, and the bacterium stops and rotates. So the fancy word is tumble. And this means that if you want to study the bacterium, a lot of times what you use is essentially a random path through the bacterium's environment. The bacterium will swim for a little bit, then it will stop and it will tumble. It will have some new direction, and then it will travel in that direction, stop and tumble. And this is called the run and tumble model. Now, this model is different to the video that I showed, and I'm going to explain why that is shortly, because in that video, it looked like they all sort of were moving in the correct direction. But for now, I want to try and model this run and tumble approach. And the algorithm, or the method that we could say maybe the bacterium is using, is that over some number of steps, I'm going to pick a direction for the bacterium to move in. So if I'm going to do this in 2 dimensions, I could do it in 3 dimensions, but I'm going to get a good result if I do it in 2 dimensions. So we'll pick 2 dimensions, and then at every point in time, I'm going to pick some angle between 0 and 360 degrees, and that's going to tell me which direction to go. I'm also going to select a duration of movement, and I'll say a little bit more about that here in a second. And then based off of that, of knowing which direction to go in and for how long to travel, I'm going to move the bacterium along that path based off the direction it picked and how long, for that period of time. And that I'm going to be able to very easily say, well, if I know the speed of the bacterium, which I said is about 20 micrometers per second, and I know how long it's traveling, well, that's going to tell me where to put the bacterium in the next time step when it tumbles again. The trickiest thing here is to figure out, well, how do I pick a duration of movement? And here is where we go back and say, well, what do experiments say? If you look at real bacteria, how long do they travel when they're swimming before they stop and tumble? And even though bacteria are by far way more diverse than everything that we think of as alive on the planet. So all of the eukaryotes, meaning us, our pets, everything that we see on television when we watch a nature program, plants, fungi, et cetera, all those organisms put together are nowhere near as diverse as bacteria. However, most bacteria who employ this sort of approach to exploring their environment, not just E. coli, an enormous collection of bacteria, are going to tumble about once every second. And so there must be some reason for that. It's a conundrum that you would have so many different species of bacteria, but they all have sort of the same approach to how they explore their environment. And so it's odd, I'm going to come back to this here shortly. It's odd that they would tumble exactly every 1.0 seconds, but it's actually not exactly every 1.0 seconds. Sometimes they tumble more frequently, but sometimes they wait a little bit longer. So sometimes they wait 2 seconds, sometimes it's a half a second. And researchers have found that a good way of fitting this is using what's called an exponential distribution. So on average, we're going to tumble every 1 second, but you're going to be more likely for it to be less than a second. And then occasionally you're going to have your wait time be more than a second. So more often than not, you're going to be less than a second. Sometimes it's going to be like 2 or 3 seconds. And we can write some code to simulate this. So what we did here was we started a bacterium at this point here, which is 0, 0, just at the origin. And we have a background gradient. So this is trying to model the circumstance where you put like a sugar cube here. And so what happens is that the concentration, if this is like an aqueous solution or something, the concentration of that sugar is going to be at its greatest at the sugar cube itself, but then it's going to diffuse, and the gradient is going to be, a little bit less glucose as you get farther and farther away. So what happens is when these 3 points are the finishing points of 3 bacteria that are employing this algorithm, this random walk. And what we see is that, well, they're going in these arbitrary directions. And as you might expect, it's weird to think that they would ever make it here unless it was just random chance. So there's nothing that's guiding the bacteria in that direction. As I said, the ending points here are the red points. And there's an extra fact here, though, which is to say we need to know more about the biology, because this can't be how the bacterium is actually exploring its environment. And there is one tiny extra fact that winds up being very irrelevant here, which is that how long the bacterium runs between times that it stops and tumbles is not actually just a random number. It depends on the change in the attractant concentration, meaning how much glucose that it detects, but the change that it experiences between the current time that it's testing and sort of what it has seen very recently. So it's able to detect, in other words, if it's swimming in the correct direction. And what this means is that if the bacterium is traveling in a direction that is heading not necessarily exactly towards the goal, but let's say, for example, a bacterium is here and it's going in this direction. If it's going in this direction, well, the amount of glucose is getting redder. So we're increasing the amount of attractant for that bacterium. And so what that means is, well, if the bacterium can detect that, it is more likely to run for longer. It's going to carry on and continue and not tumble. It will eventually tumble, but if it's going in an increasing direction, then it's likely to run for longer before it stops and tumbles. And so what we can do is we can take that fact, which is confirmed by experiment, and it's understood literally on the level of molecules and how the bacterium is taking things, surface proteins, sensing glucose in its environment, and making a change internally that changes how it operates the flagella. We can take all that biological knowledge, and then we can make a little bitty change to the algorithm that we had before. So we're still going to say run and tumble, run for some period of time, and then stop and pick a new direction of travel. So every so often, we're going to pick a new direction of movement and move the bacterium to the new position. But we're going to change how we selected this duration of the movement. So we're going to say, how long do I need to travel? And what we do instead is, and I could give you a formula for this, but I'm just going to say how the formula works, is that the bigger the difference between the concentration of glucose or the attractant where I am now and where it was previously, I call that delta L. If that's a big difference, which means I'm going in the right direction, I'm going to keep going. So I'm going to walk for longer. That's going to increase the amount of time that I'm running before I stop and tumble. And that means I can then go back and change my algorithm. And so this time we have 3 bacteria that are simulated here. And when I've changed my algorithm and I've simulated their movement, I see that, yes, they're still traveling in random directions. You see, sometimes they turn back and they're going in the wrong direction. But what is happening is, because when they go in the correct direction, they're traveling for longer. This means that even though they're not necessarily always going in the right direction, on the whole, there's almost this magnetic force that's generally sort of pulling them in the direction of the goal. So here we can measure this. And the next thing we should do is to say, well, this was just 3 simulations. I think this is probably pretty compelling, but can we do it for a lot more and measure those? And this is what I have here, which is a plot of time on the x-axis and the distance to the goal here on the y-axis. And we have our 2 algorithms, and these are 500 simulated bacteria. And the thick line here is the average distance over all those simulated bacteria to the goal. So with our previous algorithm, we started here at a little over 2,000 micrometers, and we never got closer. In fact, we drifted away on average. And this is within, this is 1 standard deviation. So this is about 60% of the bacteria are going to fall somewhere in this pink region. So most of them are not getting anywhere closer to the goal. When we change a little bit about the algorithm and just say, if you're going in the right direction or even kind of the right direction, you're going to run for longer, all of a sudden, the bacteria on average are much, much better at reaching the goal and getting near there. So as a result of this, what we're seeing is that this is a very simple algorithm in the grand scheme of things that just takes a random walk and says, if you're going in the right direction, go for a little bit longer, make your steps longer. And yet we're able to very effectively reach the goal. The way I like to think about it is that the ability of the bacterium is almost like a rubber band. If the bacterium is going in the wrong direction, then it's not going to run for very long without tumbling. And it's almost like there's a rubber band that's snapping the bacterium to stop going in that direction and move it towards the correct direction. So it's still moving randomly. It's picking all these different directions of travel that are completely random. But if it goes in the wrong direction, it's able to allow it to stop and pick a new direction. And if it's going in the right direction, it's probably not going to tumble too much. You can see this rubber band effect. I'm going to go all the way back and I'm going to show you the video again. And now you're going to see a few bacteria miss the crystal. No, what are you doing? But as soon as they miss the crystal, you can see them sort of slow down, and not all of them, but most of them are going to turn back around and head towards the goal. And that's because they're still traveling somewhat randomly. But if they're going in the wrong direction away from the crystal, they're not going to go for very long before they pick some new direction of travel. And that's what's going to keep them all here, mostly around the sugar crystal. Okay. Are there questions? Anybody have a question about how this works? Is there a slide that I could show more about? Are there any confusions that I can help clear up before I continue talking about this?
Any questions?
Sure, yes.
I'm sorry, but can you explain for me again about how the attractant detection work? May
What works? I'm sorry, I couldn't quite hear.
can you explain again for me about how the attractant detection work? How it work?
This is where we need another hour. So the cell, the bacterium itself, you're saying, how is it that it's able to detect attractant in the first place?
Right, right.
Yes. The bacterial cell itself has surface proteins. So the surface proteins cover the cell, and there is part of the protein that is in the environment and part of the protein that's on the inside of the cell. Each of these receptor proteins, you have receptor proteins that can bind to a lot of different molecules in the environment, but you're going to have some receptor proteins that are able to bind to a glucose molecule, meaning other molecules in the environment will come in and bounce off, but the glucose molecule, it will form a bind. When that happens, it changes the part of the protein that's on the interior of the cell into a different state. And when that happens, it causes a cascade of chemical reactions inside of the bacterium that wind up being like what are called like phosphorylation reactions, where a phosphoryl molecule gets handed from one molecule to another. And this sort of cascade of information exchange eventually makes it all the way to a protein that is on the flagellum on the inside. And that causes this to change its behavior and causes that flagellum not to rotate clockwise. Okay, so its default is to rotate counterclockwise and to swim. And when you detect, but it's occasionally going to switch. And when you detect the glucose, that cascade of chemical reactions is going to cause that process to stop. So it will no longer occasionally switch because the signal has been detected and that signal got passed within the cell and caused a change. This happens with all of our cells all the time. But it just happens to be very well studied within this particular bacterium. Does that answer your question? Give me a thumbs up. Kind of, sort of. Okay, good. Excellent. Anything else for now? Yeah. Excellent.
Can you move back to the video?
Sure. Okay.
Sorry, but I wonder that how do they know they are walking the wrong direction and move back to the sugar?
Yes. So
They can't
because
smell or anything else.
they cannot smell. No. The sense that they have is that they have proteins on the exterior of the cell. And those proteins are able to detect if there's glucose present, but it's also able to detect how much the concentration of glucose has changed in recent time. We're going to be deep into biochemistry for me to give you a complete answer. And I actually have a website where we explain all this in detail because it's going to take an extra 15 to 20 minutes on how it's so good at detecting a change in its environment. But it's able to detect that change based off, because what happens is the glucose molecules are coming in and binding to your surface proteins, and sometimes they release them. So that's a good thing because it means that you're not remembering, oh, I bound to one glucose molecule 15 years ago. You store it, you hold on to it for a little bit of time, and then you release it. And because you do that, it allows the bacterium to detect a change and keep track of how many of these molecules have it has bound to in a period of time. Because of that, it means I'm able to say how much glucose is there now compared to a previous time, and that causes an internal change within the cell that changes how the flagellum rotate. Okay?
One more question before.
Good. These are excellent.
Hello, can I ask you that if in the environment the concentration is not that gradient, I mean, like if you have several spots of glucose and it's much more complicated than one glucose.
Yes.
The bacteria to keep that speed or slowing down or something.
Well, if you have multiple, it's a good question. If you have multiple sugar cubes, then the bacteria are just going to move to whichever one is nearest to them, right? So it doesn't necessarily make their problem harder. When the bacterium encounters a sugar cube, it's very happy, right? Because it's found its food source. The one thing I will say on top of this, in terms of the fact that the system itself is very complex, is that the bacterium is also good at detecting slight changes in the glucose concentration or its attractant concentration across a huge order of magnitudes. So it's not like it has to have a lot of glucose. It's not like it has to be in an area where there's an enormous amount present. It's also able to detect a very, very slight change so that it might be very distant from a sugar molecule or sugar cube, or that glucose source might actually not be very big. It's still able to move in the direction of it. So it's like a very complex biochemical system.
Thanks so much, Philip.
Okay, excellent. Excellent. Those are great questions. I know I'm going to go past the hour, but so the one final thing I wanted to say about this is that there is the one question that researchers might have had was, why is it that it tumbles if it does not detect any sugar? So it just in the absence of any attractant, why is it that its default tumbling frequency is once every 1.0 seconds? And there's a famous quote from Dobzhansky that says, nothing in biology makes sense except in the light of evolution. So there must be an evolutionary explanation for why the bacteria would travel only for about a second and then tumble. Why not do it for 5 seconds? Why don't you have some, in life, we see huge amounts of variability. So why do we not see some bacteria that tumble for 0.01 seconds and some that tumble every 10 minutes? And we simulated this process and we said, okay, well, let's change the default tumbling frequency. That's when I don't detect any change in my attractant. And I'm going to set it to 0.2 seconds. So the bacterium is only going to swim on average for about 0.2 seconds, and then it's going to stop and tumble. That means that the rubber band is very tight. It means that the bacterium is not actually able to travel for very far before it tumbles. And when you look at the simulation, you see, even though you would like to go in this direction, the bacteria are constantly getting pulled to tell them to stop. And when we change that to 5 seconds, then the rubber band is very flexible. If they could swim past the goal, for example, and their default tumbling frequency is not going to be very frequent. So because they can go past something for 5 seconds before they turn around, it means that they can, run past the goal and maybe miss it. So you see these bacteria that if the goal is here, this bacterium is basically there, but this bacterium is still swimming around and is not really that close to the goal. And so what we have is, this is my Goldilocks picture to say that what has happened is a sweet spot parameter. You don't want the value of this tumbling frequency to be too small. You don't want them to be tumbling all the time, and you don't want the bacteria to be tumbling very occasionally. What you want is to tumble about every 1 second. And we made a plot of this. So we said, let's change all the tumbling frequencies. Let's look at what happens when they tumble every 0.1 second on average. And we saw that they will reach the goal, but this is the average bacterium. They are taking forever. This is our time on the x-axis. Those bacteria are taking forever to reach the goal. If you look at 1.0 seconds, it's this green color here where they tend to head towards the goal and then they stay there. So they not only head towards the goal pretty quickly, but they also remain near the goal. If you look at tumbling every 10 seconds, that's where they're going towards the goal, but then they're sort of swimming around it. So they're not able to hone in on the exact location of the goal. And what's amazing about this is that this is all from a very simple algorithm where we just simulated 500 bacteria in each example and said, how good are they at getting close to the glucose crystal that we put in the center of the field? So it's pretty cool that bacteria have somehow evolved this mechanism where all the biochemistry takes place inside of it, but that biochemistry is very finely tuned in order to perform a very specific function to help the bacterium find and remain near the food as effectively as possible. Now, computer scientists look at this problem and they say, you know what, I'm not sure, like that's a neat visualization, but it's not what classic computer scientists do. They state a problem, then they come up with an algorithm for it, and then they, say, I've solved my problem. But this is not a hard problem to solve if we're, intelligent beings. Just move in the direction of greatest increase. So at any point in time, like figure out which direction is the best way to go and move in that direction. But what nature evolved here is to say, we have a system that doesn't have smell, it doesn't have taste, it doesn't have complex senses, and it doesn't have intelligence, but it's nevertheless, evolved this approach that's very robust, and it relies on random walking, but there's no communication among bacteria, there's no intelligence here, but all it does is say randomly walk, but if you're going in the right direction, let's keep going in that direction a little bit longer. And that is a great example of what we call an algorithm in nature, which is an approach for solving some problem that might not be perfect, but that's inspired by nature and that often nevertheless is beautiful. Okay. That's end of part one. Are there any more questions on part one? I have two more stories to tell, so I don't want to run too long, Pavel. I know you're speaking today too, so.
Yeah,
okay.
then let's move to the next one.
Okay, good. Okay. So as I said before, slime molds are foragers. They eat most of them eat decomposing matter in their environment. Let me make sure the sound is off in this. They explore sort of outward. And as you can see, the slime molds here are happy to explore a lot of different regions in their space. But over time, and this is a real slime mold on a board that has food sources placed on it at different points in time. And over time, what you're seeing is that the slime mold is evolving into a network where regions that are not found in the network, like these areas up here, these regions are not part of the network because there's no food up there. But there are food at other places. And so the slime mold has been able to, over time, develop this network so that it's able to transport food that it finds at one position to other points on the slime mold. It's sort of a beautiful picture that it has. And what we want to do is to say, how can we understand the approach that it's using for, again, exploring its environment, but then doing something that's more sophisticated, I think, than what the bacterium is doing. What happens here is that the slime mold encounters a food source, and I want you to think of that as like a hub. That's like your bus depot or like a train station where you might be able to go to different places. And that hub is able to send the resources that the mold encounters to other parts of the mold. What it does also is when there's a lot of food, it's going to reinforce the pathways connecting those two really good hubs. So these are like big cities or important places in a country to build depots. It also gets rid of things that are not very necessary. So it starts with some network, and then it says, you know what, this connection from place A to place B is actually not that important because it's not helping the mold send resources, send the resources from the food that it's encountering in its environment to other parts of the mold. But it's not clear at all what problem this slime mold is solving, right? And so one way that we might model this is using something called a Steiner tree. So a Steiner tree says, okay, we're going to take endpoints in two-dimensional space, and then we're going to construct this tree called a Steiner tree that contains all n of these points. And the Steiner tree includes the points as nodes, but it also minimizes the sum of edge lengths in the tree. In other words, the total number of connections in the network, the lengths of all those connections, when you add them all up, is as small as possible. And so this is almost as if, like, think about you have a collection of cities, and we want to build a rail network connecting all these cities. And we want to use as little rail line as possible because we have finite resources. And the slime mold's kind of doing this too. So I'll show you an example. We've got 4 points, and they're the 4 corners of a square. Let's find a Steiner tree of these points. So remember, the Steiner tree is a tree, so it's not going to have cycles, but it's going to be a network that connects everybody, all the nodes somehow. But we also want the edge lengths, when we add up the lengths of these lines, we want those to be as small as possible. And what probably the first thing, if you look at this and say, well, how could I connect all these points so that they're all connected to the network? A very natural thing to do would be something like this. Let's make one of those cities like a hub, one of those points a hub. And then I'm going to connect it to all 3 of the other ones. So I'm going to have the 2 sides of the square, and then I'm going to have a diagonal. So it's going to be possible to reach any node from any other node. So the network is connected. But what the question is this the best one? And we could add up the total distance of all these edges. So if this is a square with side lengths 1, we could say, well, this is 1, that's 1, and that's the square root of 2. So the total distance of all the edges is 2 plus the square root of 2. And then the question is, can we do better? And in this particular case, we can. A lot of times students will tell me, well, okay, you connected the nodes like that, but I could have connected them like that. I could have just said, let's do 3 sides of the square, and that way we're still going to have a connected network, but the total distance here is 3, which is less than 2 plus the square root of 2. And then we say, can we do better? And usually when we have a lot of time, I'll ask students, can we do better? And they'll all agree, we can't do better. I can't come up with anything that's better in this particular case. And I love that because it means I can trick them, because the correct Steiner tree for these points is this. So you can actually prove mathematically that the best way of connecting these points into a network with using as short a total collection of edge lengths as possible is this network. And why it's tricky is that the Steiner tree actually adds 2 new hubs, 2 new depots here. And when you do all the math at the appropriate angle, all these edge lengths wind up being the square root of 3 over 3. And this edge length winds up being 1 minus the square root of 3 over 3. And when you add all that up, you get 1 plus the square root of 3, which is a little bit less than 3. And it winds up being the best answer. So that I show students that because it's a simple example. The answer is hard and not obvious. And so we must be like not doing something like this. The question is, how is the slime mold actually exploring? Because the slime molds don't know that this problem that I gave you is very challenging. So there is no algorithm that is fast that any computer scientist has ever come up with to solve this problem for an arbitrary collection of endpoints in 2-dimensional space in a fast way. But the slime mold is fine. It doesn't mind that this is a very challenging problem to computer scientists. The slime mold still is able to find food in its environment. And so the question is, well, why don't we see what it actually does to explore? So when you saw the start of the video, I've got some stills here. One of the things I made sure to point out is that the slime mold was exploring in this direction, sort of left to right here. And it's exploring the entire space really well. So how we think of this is that it's a very thin lattice, so a very thin network with a lot of coverage. So there's all these very thin edges that are expanding outward on the outside of the slime mold. So that way the slime mold's able to basically encounter most points as it spreads out. But as it spreads out, it wants to remove a lot of these little connections. So when it finds food at 2 points, it reinforces the connections between those points. So when we go from here to here, you'll see you got rid of a lot of unsuccessful connections because you didn't find food in this area, but you're also going to reinforce like this big vein. This is like a really important highway or rail line because it's connecting a lot of food sources. It allows the areas that were not very successful, as you see, to sort of die out, and that produces the final network that we see. So we can mimic this. But what I'll say is we can mimic it by saying, all right, we can build, write a computer program to build a finely meshed lattice that covers a space, and then we're going to repeat a bunch of steps, some collection of times. We will pick 2 arbitrary nodes as source and sink. And again, you may have questions that are related to the biological study and all the lab experiments that went into this. This would be that much more complicated because it's not quite as well studied or simple a model as the bacterium that we looked at. But we'll repeat these steps a number of times. We're going to pick 2 random nodes. And what we're going to do is we're going to try and push as much flux or as much pressure from the source towards the sink as we possibly can. And what we're going to do is along the way, so this winds up being a linear algebra problem. It winds up being like university level mathematics, but we can write it all out and say, solve this out so that the flux in and the flux out balance at every node along the way. And we're thinking almost of pushing water through this entire system. And then what we're going to see is when the water flows through the system, okay, where are the tubes? Where are the connections that basically didn't have much water going through them? So when you go from point A to point B, some of the tubes are going to have a lot of water flow through them, and some really aren't because of the nature of how the network is set up. And you can compute this, as I said, with a little bit of university level mathematics. And after you've done that, you're going to say, well, where did the water flow? And wherever the tubes were that had high flux, we're going to say, reinforce those. We're going to make those tubes thicker. They must be important. And if you didn't have a lot of flow, then the tubes are going to get thicker. Thinner. Those connections are going to weaken. And if we do this over a series of steps, then over time, edges that are very important are going to get reinforced and are going to get thick, and edges that are not important are going to get very thin and then eventually die out. So researchers said this is basically an algorithm that we came up with that's inspired by looking at how the slime mold behaves. And so let's do this for a real problem. There was a fun scientific paper from about 15 years ago where they said, we are going to put a slime mold on a plate and we're going to give it food sources at different points. And those food sources are going to be the exact locations of cities with high population areas around Tokyo. And then they said, well, it's not possible. So like an area like this, I think is like a very mountainous region, and it's not possible to build a train through the mountains. So we're not going to allow the slime mold to have any light here. The slime mold needs to have light as well as food in order to grow. So we're just going to keep this turned off. So they did this very clever thing of putting light where it actually is feasible to connect points, and then the points themselves are food sources for the slime mold. When they did that, what they found was that the slime mold evolved this network here on the left, and the real Tokyo rail network is shown here on the right. So it's not identical, but there are some, I think, remarkable similarities about the network that the mold evolved and the network that humans created. And then we also can look at the one that's simulated based off of the algorithm that I told you about. So we can take what it seems like the slime mold is doing, and then we can create an algorithm that, updates the flow along these edges as we go. And that's what this network here is shown on the left. And they're all a little bit different. And part of the reason why is that there's really 2 things here that we want from any network. for the slime mold or for the real rail network. We want the network to be small. We don't want to use a lot of rail, right? We don't want to have many, many connections because we only have so much budget. But we also want it to be that if one line from one city to another, if there's construction there, right, or some reason that a train breaks down and you can't use that line, well, we don't want that to mean that it's completely impossible to go from one part of the area around Tokyo to the other. To say that that would completely disconnect the network, a cow steps in front of the line and that's it forever. You have 3 months before you can make it from one part to another. So that's a real term that's called fault tolerance. You want it to be so that you can actually remove a few edges from your network and it be okay. So for example, if we removed this edge from the slime mold network, it would still be possible to go from these cities to each other. We would just go around. And these two things that we want are conflicting. You want to not have a big network, but you also want it to be that you can remove some edges and it's still going to be possible to travel. When we look at the simulated networks, they're more efficient. So if you look at this network compared to the real network, as well as to the Tokyo network, what you see is fewer edges. So the simulated network actually did a good job of having a very tightly compact network of travel, but it does a bad job with respect to the fault tolerance. So it is possible to go in and remove just a few edges and create havoc over here where it's not possible to travel between edges. And this network here has more edges and is just a little bit more protective of the fault tolerance. And if you wonder why this might be, it's that because of evolution, the slime mold needs to have a few more edges so that, for example, some predator doesn't come in and eat part of the slime mold or somehow prevent it from being able to travel between a couple of edges. So for that reason, there is some complex aspect of the algorithm that the slime mold is actually taking that says when you look at these two conflicting things that we want, the fault tolerance is a little bit more important than just having a small network size. So I thought this was a fascinating sort of study because what they did was connect it to a real rail network, but also look at what the slime mold actually evolved and compare that against how they thought the slime mold was behaving and noticing that you got something a little bit different in each step of the way. Are there any questions here before I continue? Yes, Pavel. Okay.
Yes, questions.
Oh, I'm terrified of this. Okay.
I didn't understand one part of the algorithm. So
Okay.
when you push flow from the source to the descent,
Yes.
the capacities of all edges have to be defined, but it's not clear what are the initial capacities, how they're defined.
When you start with the thin lattice, they're all equal.
Oh, in the beginning, they're all equal. Right?
They're all equal.
Okay.
Yes.
It makes sense.
Okay.
How much of maybe the slime mold network do you think, attributable to just chance? I mean, is it if you have did the experimenters do the slime mold thing with more than one dishes of slime mold, and did they always produce the same network, or is it pretty?
No. So, I mean, there's by chance, what I thought you meant was its ability to create edges. The ability to create edges connecting food sources is not that random because it starts off with a pure random exploration, right? Like this stage, when it's just moving in this direction, it's trying to cover as much of the space as possible. So from that perspective, the first step of the algorithm is random. I think your question is, if we ran this experiment a million times, what is it? How similar would the networks be, right? Over time. Yes. And that I actually don't know the answer to that. And I would think that's a fantastic question, which is this is in the same way that I ran 500 simulations before to show you that, the bacteria truly were able to reach the goal. It was robust to having different bacteria. Even though they were applying random logs, they all got there. The question would be, how is it that these networks change if I do this for 100 or 500 different slime molds? And I'm sure they have done that, but I don't have the answer immediate. I think that's a good question.
Okay. Just one more questions.
So in your personal opinions, the slime mold network or the Tokyo Rail network, which one is more fault tolerant? And is this slime mold network algorithm can be modeled to the network of transportation? It's not only the rail network, because it's more similar that way to me.
I think they could. I think this is the one that they picked, and I don't know about downstream studies and looking at other transportation networks. Your first question was really good, which is if we look at this real network and look at the example Tokyo Rail network, how does their fault tolerance compare? And I seem to remember that the fault tolerance of these networks is about the same. It's be a different direction today to say, well, how do you define fault tolerance exactly? But there's mathematical formulas for doing this. If I have a network, I can have a function that looks at that network and looks at all the connections and measures the fault tolerance of it and says how fault tolerant is it. These two networks, when you look at that metric, those two networks wind up being about the same in terms of fault tolerance. But when you compare it against the simulated network where they said, let's see what we think the slime mold is doing, this network winds up being always less fault tolerant. Okay, so the numbers wind up being about the same here, but very different when we look at this other network.
Thank you.
Excellent. Okay, Sean, should I continue?
Please.
Good. Okay, these are such good questions. Okay. Finally, I want to talk about one more organism, which is Drosophila. Drosophila is the fruit fly and has been the model organism that we use for genetic studies for the last 120 plus years. So the fly itself is covered in sensory bristles. And when the fly is developing, so at the point that the fly is just a fly embryo, the fly has clusters of cells, and all these clusters of cells are near each other. And some of those get chosen to be sensory organ precursors. I'm just going to use the term SOPs. So as the fly is developing, you're going to have a mass of cells that are all next to each other, and some process goes on that picks some of these cells to become SOPs, and those cells are then going to become bristles. So those are going to become the sensory bristles that the fly has on its body. But it doesn't have millions of them. You can see them here, right? You can see all the individual little bristles that the fly has. And so some researchers said, can we study the process by which the cells are getting chosen to be SOPs? This is as unintelligent a system as you can get. The fly is not directing, in the same way that you say, why is it that like the hairs on my arm were chosen at different points? Well, what process within the development of the cell chose all those exact points to put bristles on them? There's a little bit of biology we should know then, which is if you start with your original sort of cluster of cells, any of those in theory could wind up becoming an SOP, which means they're going to be a bristle. So any of them could win this lottery of sorts. But when one starts to appear, so if this cell, for example, starts to appear as an SOP, starts to the process of developing, and this often can just be, it happened to be just a little bit ahead of its neighbors in this process of development. What it does is it expresses a specific type of protein called delta in the cellular membrane. So the cellular membrane is adjacent to this cell, and it's adjacent to this cell, and it's adjacent to this cell. That protein serves the purpose of inhibiting the development of these neighboring cells. So because of that process, if this cell really starts developing as an SOP, it will essentially, for the most part, shut off all of its neighbors. So what that means is, and I think this is maybe not a completely hard and fast rule, but a pretty good one, is that you're never going to have two different cells that are neighboring, that share a boundary in terms of the cell membrane, but that are both SOPs. So here would be a valid choice of SOPs for your cells. You have a blue cell here. They're all shown in blue. You have a blue cell here it's not connected to any blue cell. You have a blue cell here, it's not connected to any other blue cell. And you have a blue cell here, and it's not connected to any neighboring blue cells. So the question that we might stop and think about is, in terms of evolution, I've got a cluster of cells. What problem, if we zoom out, what is it that the fly is really trying to do? And what it's trying to do is it wants a lot of bristles, because a lot of bristles is good. Bristles mean that it can sense its environment, but it doesn't want to create too many. That's going to create a resource issue within the organism. So you want to use as few of these bristles as possible, create as few of these bristles as possible, and you also want them to cover the body of the fly. So you have this constraint that neighbors can't wind up both being SOPs. This very simple expression of a protein turns off neighboring cells. That's a good thing, because you don't want all of your SOPs to be just up here, and then you wouldn't have any down here, so the fly would have a very dense collection of bristles just up here, but it wouldn't have any down there. You want good coverage without using many. And what we could do is say, okay, well, this actually winds up being a network problem. The nodes here are going to represent cells. So these pictures here are the network version of this picture over here. I want you to think of each cell here as a node in a network. So you can see that these correspond to like the centers of the cells that we have over here. And then you might say, well, what are the connections? Well, this cell that I pointed out before, it is adjacent to 3 other cells, and those are the 3 cells that it's connected to over here. So I have edges connecting them. And then what I want to do is I want to color the nodes of this network with one of 2 colors. Here I'm going to say blue. I might think of them as just labeling them 1 or 0, but I'm going to use colors because that's nice. So the nodes are going to get a blue color if it winds up being an SOP, and they're going to get a red color if it doesn't. All right. And then so you say, okay, this maybe thinking about this is a collection of cells, thinking about that as a network problem is going to help me understand it in terms of framing. What is it that the fly is doing? Okay. And so what it is that we're finding is what we want is an independent set. An independent set of nodes in a network means that there's no edges that connect 2 nodes. So you can see that the blue nodes here form an independent set because there are no edges that connect 2 blue nodes. So if you look at this node here, all of the nodes that it's connected to are red. Same thing here. All the nodes it's connected to are red. And if it were ever the case that like this was blue and this was blue, well, that would be bad. The blue nodes would no longer form an independent set because this node would be connected to a blue node and this node would be connected to a blue node. And so that would violate what it is that we want. Okay. So we want an independent set of nodes, meaning 2 blue nodes are not connected to each other, and we want it to be as big as we can make it. And the fancy way that we say that is we want a set that we can't make bigger. So what we don't want to do is to say, okay, well, my independent set is going to be this node and it's going to be this node, and I'm done. And I'm going to have 2 bristles, and that's all I want to do. That would be bad because then someone would come along and they would say, well, you chose this cell and this cell, but you could have also chosen this cell or this one, right? And you could have made your set bigger. You could have had more SOPs, subject to the restriction that you can't have adjacent SOPs. So if I just pick this one and this one, well, I'd have 2 blue nodes, but somebody could say, yeah, but you could get a 3rd one in there. So I want to make it as big as possible. I don't want it to be the case that I have my collection of SOPs, but it's possible to add another one. And the fancy way of saying that is what I want is a maximal independent set, meaning I have my network. I'm going to color some of those nodes blue. I cannot have blue nodes next to each other, and it's not possible to make my set of nodes bigger by finding some other node that I could add. And then we say, okay, well, we've got a problem that's stated, okay, but how is it that I could solve it? And this is another example of a problem that if you take this to a computer scientist and we think about writing code for this, the computer scientist says, I don't understand why you're studying the problem because it has a very fast solution. I can create an empty set S. So I'm going to start with no nodes, and I'm going to start adding nodes to S to form a maximal independent set. And I'm going to say, while I've still got nodes left, here's what I'm going to do. Pick any node you want, and I'm going to add it to the set. So then I'm going to get rid of that node, and I'm going to remove all of its neighbors from consideration too. I'm going to show you a visualization of this here shortly. And then S is going to be a solution. So computer scientists might say, this is an algorithm that is like almost embarrassing to write down because it's going to solve the problem. And then they're going to say, why are we studying it? All right. So hold on one second, because we're going to talk about how the fly solves it. We pick a node here. This is just an arbitrary node. This was my favorite node. Okay. That's going to be the first node that we add. Then I'm going to get rid of this node and all of the other nodes from consideration. So my set's going to start with this blue node. I'm going to pick another node that is not one of these nodes that I've already considered. And so I say, for example, I picked this one. And what am I going to do? I'm going to remove this node and all these neighbors from consideration, meaning I can color all of its neighbors red. And then I've got 3 more nodes left. So what I'm going to say as well, pick an arbitrary one. I picked this one. Its neighbors are already red, so that's good. And then I don't need to do anything. And I've got 2 nodes left. Pick one of them, pick your favorite one. I picked this one. And this is a neighbor of that node, so I'm going to color it red, and I'm done. Okay, because that's going to be my solution. I've covered every possible node. I colored 4 of them, and that's a different solution to the problem than what I had before. Okay, so this was also a maximal independent set. This gives me a different maximal independent set with a different number of nodes in it. And they're both solutions. So the computer scientists would say, we're never going to study this problem again because here's my algorithm. But nature doesn't solve problems in the way that computer scientists do. When we solve a problem in computer science, we write code assuming that we have all information available to us, meaning we are able to sit here and see this network in advance. We know all of the nodes. We know all of the edge connections. We know what all these cells are. The cells don't know that. All this cell knows is I'm next to this cell, and this cell. And it is not able to communicate really with cells in other parts of the network. It doesn't know it's part of a network. It just knows it's a cell. And so when we think about what that means is that nature is solving this problem in a manner that is distributed. All the cells have very limited information. There is no intelligence in the system, and all of the cells are doing something independently of each other. They're all doing their own thing. The researchers then said, okay, can we figure out what it is that the cells are doing? And here's after they did a bunch of laboratory work, here's what they said. This is the study of what the cells are doing. So because these are developing cells, they all start off as off. So they're not SOPs at all. And then we're going to have an algorithm that runs for a sequence of steps. As those steps go, there is going to be some probability at any point in time that a cell turns on, meaning it develops into an SOP. And that probability starts off small because on a long enough time scale, everybody would be an SOP if it weren't for their neighbors. So they're all developing, but some of them are just going to have a chance of developing faster. So again, we have the fancy word here, which is everything is based off of probability. And that probability starts off small and then gets bigger. At every point in time, the cell will say, am I on? So am I trying to activate into an SOP? If so, I'm going to message the neighbors that I have in my immediate neighborhood by expressing that surface protein that is a membrane protein that represses their development. So that essentially is trying to turn them off. And as a result of this process, if I am on, I sent this message to my neighbors and I did not receive the message from my neighbors, that means that I should become an SOP because no one else around me is trying to turn me off and it's okay for me to turn on. So I'm going to continue keeping all of my neighbors turned off. I'm going to continue sending this membrane protein to repress them, and I'm going to become an SOP. If I did receive a message, this means you have 2 conflicting SOPs that are next to each other, and they're going to turn each other off, and one of them is going to eventually have to be chosen. Then there's a final step here, and again, I'm giving you the overview of the whole thing, but I'm going to then show this to you here and with an example, and it's going to make it a lot clearer. If I'm off and I got a message from someone, that means that I've lost the game. All right. That means that one of my neighbors is repressing me and happen to turn on. So I'm going to remain off permanently, and that the algorithm's going to stop for me because there's no reason to continue. I'm not going to develop into an SOP. So let me show you an example. The first step is to turn on some collection of these cells. And by some probability, as I said in the start, it's not going to be pretty low. But say, for example, that these 3 cells turned on. What they're going to do then is try and broadcast the message to their neighbors and say, hey, I'm on. So if 2 neighbors that are next to each other broadcast to each other, that's going to serve to keep them both repressed for this time step. So they'll sort of try to turn on, and then they'll express the protein to each other, and that's going to keep them off. This cell, however, is different because this cell turns on, but it didn't receive a message from its neighbors. And so that cell here is going to survive. It's not repressed. It's going to allow itself to grow and develop, and it's going to become an SOP, which means we need to tell its neighbors that they're going to be off forever. So this cell is now going to repress its neighbors. It's going to develop enough to be an SOP, and the neighbors are never going to turn on. Then what do? We say the next time step goes by, and in the next time step, say that we have 4 cells that turn on. These 4 cells here are shown as blue, and we say, okay, well, which of these cells are neighbors of each other? In this particular case, these 2 cells are neighbors, and these 2 cells are neighbors. So we're not going to keep these on. They're actually going to switch off, but this node will stay on. So we now have 2 SOPs, and this cell should broadcast to all of its neighbors and turn off all of those cells. Then we simply repeat. So we've got 4 cells left that could be turned on. In the next iteration, as I said, this probability of the cells turning on is going up over time. So maybe 3 of those 4 turned on. These 2 cells that we have here are going to turn each other off. This one will stay on. It will broadcast that message to its remaining neighbor. And then we're nearly there. Maybe in the final iteration, we have this cell turned on, but this one did not. So it's going to broadcast that message to its neighbor, which will turn it off. And we have a final coloring of our network that corresponds to a maximal independent set, because you have a collection of blue nodes, which is the same number that we had before. And there's no edge that connects any 2 blue nodes. So every blue node is connected only to red nodes. Same for this one, same for this one. And that's what we want with an independent set, is blue nodes only connected to red nodes. And what was cool about this particular algorithm was this is an old, well-researched problem in computer science of maximal independent set, especially when you look at solving this problem over a collection of multiple computers. I actually have a colleague at CMU who has one of the most popular algorithms for solving this problem, and it actually has non-biological applications to it. So for example, one particular example that they looked at was mesh Wi-Fi. If you have a building and you have a collection of mesh Wi-Fi routers, you want to put those to give yourself as much coverage of the building as possible without using a lot of routers, and by trying to identify using as few as possible and putting them in locations that allow you to have coverage of their nearby areas. So it's the same general type of problem. It can be framed in that way, which is completely non-biological. The other thing I would say here is, and this is a good way of finishing up today, is that this problem, as much as any of the others, show that how we think about problems in computer science is that we know everything in advance. We know all the global information of knowing the structure of the network. And so we have total information sharing. Communication is free. So it's, if I know something about this node over here, I'm also going to know something about this node over here. And so even if I solve this problem with sort of 100 different computers, they're all going to be able to communicate with each other, and that communication among them is going to be free. And what I'm looking for is these like perfect, exact solutions to problems. But what you saw today is that you have systems that have often very limited information, especially limited information locally. All these cells will never know that they're part of a larger network. They're just going to be connected to these 3 neighbors. Communication is extremely simple. Here, the only thing a cell can really do in terms of communicating with its neighbors is expressing a protein in its membrane that serves to turn off its neighbors. And what we saw also today is that we're not solving problems exactly. Over here in computer science, we're always trying to solve problems exactly. Nature is always trying to solve problems approximately. And the fancy word that we use there is a heuristic. So a quick and dirty method that gives us some solution, but it might be a perfect solution. And the other thing that we saw throughout today is probability. So whether it's the bacterium or how these cells are developing in the fruit fly, we see that often this is based off of some probabilistic process that isn't completely determined, but that is based off of moving in a random direction or randomly turning on some subset of cells. And that's how everything works in nature, where information is limited and local. Communication is often very simple. You don't have a global view of everything, and a lot of things are based off of probability. That is the end of my talk. I want to say thank you for all of your time. I also want to make sure I have a chance to take some questions and not take up too much of your time. So Sean, thank you again so much for having me. And what can I answer?
Yes, Dr. Philip, there's one more question here.
Sure. I can't see someone. Maybe if they
okay.
zoom out.
Wow, that's a really cool algorithm. But is that what happens in nature, or is this what we think happens in nature? I mean, did they look, I'm sorry, we didn't read the paper, but I mean, did they look at the biological basis for this? And is this actually?
I did.
Oh.
Yeah, and I haven't recently, but yes, they basically hired laboratory. This is a Science magazine paper with several authors on it, and some of those people were laboratory biologists who performed experiments to verify that this indeed is what's happening in terms of the expression of these proteins.
So they looked into the biology and they found the proteins that signal this and do this. Wow.
Correct.
Okay.
Anything else?
Okay. One more question.
Okay.
Hello. I want to ask you about the solution of computer science and solution from the non about as a student about nature you have present, and you can you talk about is there any pros and cons between using each, like in for example in a mass Wi-Fi, if you use computer science solution, I think it is better because it can optimize the solution more better than the nature. And if you use the nature solution, is there anything better than?
Okay, that's a great question, and I would say two things. So to reformulate, I think what you're asking, I want to clarify. You're saying, even though, where did I go here? Okay. Even though I've pointed out how nature is solving this, and it's impressive that nature comes up with these algorithms, a lot of the time, nature is coming up with algorithms that are not actually as good as what we can come up with when we think in terms of solving a problem. And we have global information and fast computers that we can use, and lots of intelligence that we can use to solve these problems. I would say there are two examples that are worth pointing out because this is a sampling of the field. One example, neural networks. And neural networks are a simple model of how your brain works by taking a bunch of artificial neurons and exchanging very limited information, essentially moving numbers from one neuron to another neuron in this big connected network. And a lot of people for many years said neural networks are actually fun to study, but a criticism of them is that the human brain is always still smarter. The way that our brains are wired, this is a simplification of how our brains work. We're able to look at problems and solve them and come up with algorithms that are more complicated than just moving numbers around in this network. And then one day they eventually were able to use so much computing power that the problems they could solve with neural networks blew up, right? And now we have ChatGPT. We've crossed a threshold, right, with some of the large language models, as well as some of the deep learning models that we have, where we're able to answer questions that we simply could not answer before, even though the model itself is actually very simplistic. So that's example one. And example two of where this talk would be 3 hours long if I had told this story too. But there is another fruit fly story where when the fruit fly smells something in its environment, it has a very natural question that it could ask, which is this new? That's a very important question for a fly to answer. Have I smelled this before? Right? Because if I have, well, maybe it's food and I can go in that direction. If I haven't, maybe it's something to avoid or maybe something to explore. So it's a very simple question to ask. Is this smell new? Researchers dissected the fly brain. So they looked at the olfactory neurons that are connected to the nose, like going back into the brain. And they took the whole brain apart and looked at all the connections. And then they said, how is it that these connections are wired? And what is it that happens when all these neurons fire? What happens at the end result in the fly brain? When they did that, they realized that they had an approach for answering a very general question. That question is, I have a database of things and I have a new thing. Have I seen it before? Sometimes your database is enormous. So your database is like every image on the internet. Okay, have I seen this image before? Does it exist on the internet? That's a very hard problem because the internet is so large. The fly is the same sort of circumstance. The fly has smelled many things in its life, as have you, right? And so how it solves this problem is not to create a huge database in its brain. It doesn't have the space to do that. It solves the problem in a very probabilistic way, with a small number of neurons that are connected to each other. And yet, when the researchers looked at the problem, what they realized was that the fly had evolved an algorithm that was more sophisticated for the problem of trying to find, is this object in a database new when the database is huge? It was more sophisticated and faster than any algorithm that a human had ever come up with. So it gave them a new algorithm for searching a huge database to see if an object in it was new. But to explain that algorithm, I would need another hour. So I'm going to leave you there.
Thank you.
Sure. Thank you. Great question.
I think like we just have enough time for only one more question. Any
Okay.
one last question before we end this session today? Okay, so I think that we have no questions left for today. So yes, and I think like that's like the end of the session of like Dr. Phillips. So yeah, and Sung here. So we will end the session of Dr. Phillips here, and Sung will have some.
All right, so much appreciate Philippe for the excellent talk. I have to. All right, so anything you want to say, Philippe, or anyone, any questions to Philippe?
I think we handled questions, Sean, but yeah, I mean, unless they have other questions, I'm happy to answer them. But yeah, thank you so much for your time.
Any different questions, not concerning the lectures, just for career advice, for example? Any questions? No? All right. If we don't have questions to Philip, thank you for the great talk. It has been a while since I see you, but you know, we talk from time to time, but mostly on the phone.
Yes, let's talk soon, Sean. Thank you for having me. It was good to see you, everyone.
All right. All right. Thank you so much.
Bye.
Everyone, let's take a 5-minute break and we'll get into the new sections for Pavel.
Mọi người ơi, mình break chút xíu nha. Khoảng 9:35 mình sẽ quay lại phòng để bắt đầu tiết của tiết kế tiếp.